Okay, hopefully you've read the Garcia-Colling paper and done the reflection on it. One thing I forgot to mention about salience before I ended the video is that the salience can differ across species. This is something that's called species-specific conditioning. And what this means is that some cues are ignored by some species, right? And this is just due because they can't detect them due to evolutionary constraints. So, for example, cats can see color, but they don't respond to color very well. They respond to brightness much better than they respond to color. Rats respond to smells or sounds better than color because of their environment. They're both animals are nocturnal animals, so they're operating in the dark a lot. So color is not a very salient cue for them. So salience can also differ just depending on the species. That's sort of my point there. Now let's get into logical associations with Garcia and Colling. Let me break down this study for you after you've read it. Then we'll come back and talk about informativeness of the CS stimuli. So first it begins with sort of they had two lick tubes that their animals were exposed to. One, they had a CS where the water was flavored. This is what's commonly referred to as the tasty water. Another lick tube, every time, and you can rig this up, so every time the animal licks the tube, there's a click and a light. So this was called, sort of affectionately called, the bright noisy water. And in fact, Garcia and Colling's whole study is referred to as the bright noisy water experiment. Now, these two stimuli together, these are called a compound conditioned stimulus. We'll see another example of that in Caimans' blocking study in just a minute. But the key here with Garcia and Colling is these were the two tubes, and the animal had equal access to them, and they drank from them, and initial consumption from these tubes was equal. And we can see this in figure 1, and so I'm going to break down figure 1 for you. So here's figure 1, and let me label a few things for you. So the left half of this figure, the unconditioned stimulus is illness, and it was either done by an X-ray or a lithium chloride injection. And over here we had the unconditioned stimulus with shock, and they varied sort of when the shock happened. Now, as you can see, the arrow here I have, this is before the CS. So the pre here means before the conditioned stimulus. You can see that everywhere consumption is pretty equal, except it's a little bit different here in those groups. So there's nothing special about those tubes that promotes or demotes drinking from the tubes. They consume about the same. The question is, what happens a little bit later? So after the CS is consumption. So again, they get the CS and then they get both tubes, and they can drink freely from both tubes, and they just measured how much they drank from both of the tubes. Now, the key here for this is the tasty water is labeled here as the gustatory test. So that is the white bar. So what they mean by the test is sort of just giving them just the exposure to the water again. It gets a little confusing with the terminology for sure. So as you can see, after the CS, here on the left-hand side, there's a big difference in consumption, right? They're drinking more from the bright, noisy water following illness, right? And we're going to see, so let me just sort of point that out to you. So they're drinking more from the bright, noisy water following an unconditioned stimulus of illness, but the opposite is happening over here for shock. So for shock, they're drinking more of the water that tastes the same. So what's the upshot of that? Well, what the upshot of this is Garcia and Colling are showing you that certain stimuli go together in logical associations, such that the animals associated illness with taste. And because they associated with taste, they didn't drink this water. They drank from the bright, noisy water. Simultaneously, or conversely, actually a little bit better word, conversely, the reverse is true, right? So they're not associating shock or painful stimulus in the paws with taste. Instead, they're associating it with bright, noisy. So maybe like lightning, and all animals and are sort of or predisposed to associate sounds and flashes of light with painful stimulus, right? And so the idea is here through some evolutionary constraints, some stimuli are easier to associate than others. If all stimuli were easily associated, you wouldn't see these differences. You would see them avoid both tubes because they experienced two unpleasant, unconditioned stimuli. So they would just avoid them altogether. But you're not seeing that, right? You're seeing these different patterns of behavior. So that's the Garcia and Kohling telling us that some stimuli go together. The other thing is that some stimuli through experience can be more informative or in some cases less informative than others. And so a couple of experiments demonstrate this. The first that I'm going to talk about is Caimans' blocking experiment. And so what Caiman did is he had 16 trials of a noise, which was the conditioned stimulus, followed by a shock, which was the unconditioned stimulus. And of course, the animals are going to freeze to that. Then he followed it with 8 trials where the noise plus the light, this is now a compound stimulus, just like we saw before, followed by shock. And so what Caiman was interested in is like what kinds of conditioning developed to these conditioned stimuli. So the noise, the animals froze, so there was a conditioned response. But interestingly, the light, there was no conditioned response. So in other words, the conditioning of the exposure of the noise and shock relationship blocked conditioning of the light and the shock relationship, right? So, and by the way, you might get hung up on the number of trials. This is just one example of many studies that he ran. It doesn't really have anything to do with it. You can change the number of trials, and the reality or the outcome is basically the same. Now, he did follow up with a quick study that initially the animals were presented this compound stimulus, and you do get a conditioned response to both of them if they're initially associated with the unconditioned stimulus. So the common explanation for this is that the pre-existing relationship here, they're first exposed to the noise and the shock, blocks conditioning to another stimulus because the light, if you think about it, adds no new information, right? The light is never presented with the shock without the noise. So there's no new information added here. So there's no conditioning that gets associated there because there's no new information. So again, that tells us that a CS has to be informative. In the case of the light with the blocking study, not informative, doesn't get associated. You also see this, remember in the Garcia and Kohling paper, I told you to pay attention to habituation. So they habituated their animals to the cage, right? So the context or the cages can provide lots of cues. So one thing you want to do is have them exposed to the apparatus without anything happening, so they habituate to that. That way they don't associate the cage with whatever's happening. Instead, the experimenter can isolate whatever the conditioned stimulus is, right? So we know that just contingency, contiguity alone don't make a good conditioned stimulus. It has to bring some information to the animal, otherwise it's not going to get associated. So another example of this in effect is something called the CS pre-exposure effect. And here's how it goes. One group is exposed to the CS, where you just present the CS alone for several trials. Remember, it's a neutral stimulus, doesn't do anything. And you compare their learning to a control group where there is no CS pre-exposure. So now you try to learn or form an association by pairing a CS and a UCS. And what you find is the pre-exposure group has some slower learning, latent learning or latent inhibition. The learning is slower. You have a flatter acquisition curve. And the reason for this should be pretty obvious to you, right? If you're exposed to the CS alone, you're going to habituate to that. You're going to learn to ignore that stimulus. And then when you learn to ignore that stimulus and the contingencies change, you can learn a new association. You can learn that CS matters, but it's going to take you a little bit longer. It's going to be a little bit slower, right? So not all CS and UCS stimuli are created the same. The last thing I want to talk about here in classical conditioning is the concepts of generalization and discrimination. So the generalization is when a conditioned response is elicited by other similar stimuli, right? So for example, and in this example I'm going to develop, let's suppose that we have a green light and we pair that green light with food. As you know from like Pavlov studies, we present that green light, eventually the dog's going to salivate, right? It'll learn an association. If we then inserted another light, like a blue light, chances are the dog would then generalize and salivate to the blue light, even though it was never associated with the food, because blue and green are pretty similar. stimuli. Discrimination, though, on the other hand, is when two stimuli elicit a different response. So, for example, the green light elicits a conditioned response and the blue light doesn't, then we would say that the animal discriminates between the two stimuli, right? Can tell the difference or discriminate between the two stimuli. Now, this is the key to understanding experiences of animals. So you may have heard that dogs can't see color. They see in black and white. Well, that's not exactly true. Dogs can see color, but their color perception is different than our color perception. And what we can do is you might want to know, like, how do we know this? How do we discover this? Well, we can do classical conditioning experiments that tell us that this is the case. So what we could do is something I was mentioning just before. So we could take a light like a green light. We're going to call this a CS plus because this light is associated with food, the unconditioned stimulus, and we're going to measure the unconditioned response, and eventually we're going to test the green light for a conditioned response, which would also be salivation, right? And then we also have another similar stimulus, CS, we're going to call this CS minus, the blue light, because it's never paired with any food. And what we can do is we can plot the probability of a conditioned response over the number of trials. And remember, a trial here is a CS-UCS pairing, followed by the test trial to see if there's a conditioned response, right? You have to probe it to sort of see if there's a conditioned response. So what would happen is you would pair the CS and the UCS for several times, say like 5 times, then test to see if there's a conditioned response, and then you're going to plot whether or not they have a conditioned response or not. That's the idea. So let's take the easy case. Let's plot what happens with the CS and the CS plus the green light. So as you might imagine, with several UCS pairings, you're going to get this nice acquisition curve here, right? It's going to build up over time. So this is just like what we talked about when we talked about acquisition. You'll get that effect, right? The question is, what will happen when we test the blue light? So you can pause this for a second and think about, like, what do you think will happen, and see if what you think will happen actually happens. You'll probably be a little bit surprised. So this is what would actually happen. So we present the blue light occasionally. Now what's going to happen is early on, the animal is going to generalize. So these lines should be pretty close together. So it's not going to be able to tell the difference between the blue light and the green light until it has a lot of experience. Once it has a lot of experience, and what you would do is you would intermix these CS, UCS pairings, CS test, and CS test probe trials. You're going to mix them all in together until you see sort of stable responding. And after a number of trials, eventually the animal is going to sort of determine or discern, hey, the blue light means nothing, right? And its response is going to extinguish, it's going to drop off to where you don't have a probability of a conditioned response. But early on, the animal is going to generalize. So in here, you're going to see some generalization. But later on, you would see the separation. That's discrimination. And so if the animal can discriminate, can see the difference between the two lights, it will discriminate. It'll have a different response to each of the stimuli, and we know the animal can see those colors. In doing these tests, we know that dogs cannot discriminate between green, yellow, orange, or red. They all look the same to the animal, so perhaps like a brownish like color, but they can see blues and greens and shades of gray and things like that. So their color vision is very limited, but it's not necessarily exactly black and white. And so that's generalization to discrimination. So let me ask you a real quick question to sort of finish this. Why do you think we generalize? Why do you think the dog automatically responds to the blue light in the same way? So think about that for a second. Maybe you can pause the video and see if you can come up with the answer. But the answer is there are no perfect stimuli in the world, right? If we only responded to perfect stimuli, when we saw versions that were kind of like that, think about eating an apple. If you only associated one specific kind of apple with being food, you'd miss out on other apples. That's not a good thing, right? So what we want to do is we want to respond similarly to similar items in our world. So we're going to generalize until we accumulate different information, like in the case with the dog here, until we accumulate different information and decide, or learn, I should say, until we learn that there's a different response that's required.